Well, I started my career working on the population dynamics of insect pests, and this is a tomato hornworm. The white things on its back are wasps that control them, and I was looking in the little one at the top. There's a very tiny wasp of the wasp. So I was looking at the population dynamics and how these play a role. But after 2 or 3 years, I decided I would enter into a new field. They actually hired me looking like this. It's called chemical ecology, which is studying naturally occurring chemicals and how they govern intra and interspecific communication systems in the insect world. And one of the areas I work on is mating and the role of chemical cues in choosing a mate. And also importantly, as you can see in the top one, now how you avoid a mate that is no good at all. And it is extremely important because there are no lawyers in the insect world. So if you make a mistake, you pay for it. Another area that I work on, in particular in chemical ecology, is insect migration, using reproductive biology to study it. One of the insects I work on that is not a strange tree. It is a tree in Mexico with over 1 million monarch butterflies. And we're working to know how do they know where to stop. And we think they leave chemical cues. But my sort of Drosophila white rat is this insect here called a true armyworm. And using pheromone traps, which is the one at the bottom, and light traps, we monitor the seasonal biology of these insects, which come into Canada every year. And the question was, are they residents or are they migrants? My goodness. And when you dissect the females that you find in the summer, when the days are long and the temperatures are warm, you find the ovarian development is full, she's full of eggs, and about 98% of them contain this little thing at the bottom called a spermatophore, which is the sperm and the nuptial gifts given from the male. However, when you dissect the ones that you catch in the fall, they're little fat things and they have no ovarian development. And it had been suggested that insects that don't like the habitat will migrate in response to changes in ecological conditions. Short days, cold temperatures, tell rich Canadians, go south. Well, insects are the same. So then we took this animal into the lab and we looked at the effects of temperature and photoperiod on female calling behavior. The little gland exposed at the bottom is a receptive female emitting a sex pheromone, which is like a perfume, telling the males of that species, I am ready to mate. Doesn't mean she will, but it means the male can try. And at the same time, at different conditions, we test the response of males in a wind tunnel, reared under different conditions, to see how they respond. And at the same time, on your left, you can see the brain of an insect, and we remove the glands that control the hormones that affect maturation. We can get an understanding, and we came up with this model. It's too cold in Canada in the winter. They overwinter in the south, but it's too hot in the south. And the interesting thing, short days, cool temperatures are the cue to get them to move out from the spring, going north, south in the fall, and the winds are in the right direction. But I don't like just sitting around, so I found non-migrant populations. One in the archipelago, the Azores that you can see here. It's really nice to go there. And the other archipelago, of course, is the Pacific one, Hawaii. I go collect the moths, my students do the research. But it's sort of fun that way. And it's extremely important. And now that we're doing comparisons of the morphology, the wing surface area, the muscle structure, looking at whether the ones in the fall that are migrants differ in the number of mitochondria, the enzymes, and at the same time, the utilization of fat, because there's a cost to migration. And we're predicting that migrants will do it differently. And of course, the other aspect that helps us is that they use day length and temperature. So we're interested in the effect of climate change, because day length isn't changing. If it does, we got more problems coming. But the temperature is, and they're getting ambivalent cues. Stay, leave, and we're looking to see how this might affect them. But the other side of my research, of course, is applied, because insects cause about 30% of our crop losses, and we're trying to find ways of controlling them, avoiding what has always been the first resort rather than the last resort, and that is the use of insecticides, where we basically just spray everything. And we know that there's a problem, resistance develops, The pollinators, like the bees, get affected, other organisms like the frogs and the birds, and occasionally human beings. This is an undergraduate, we could replace them quite quickly, but normally we are very careful, and this isn't an insecticide, it really is calibration. But using pheromone traps, we can actually monitor populations, and we look at the number of counts per week, for example, and we can say to a farmer, this time you have no problem, don't bother to spray. Or, wow, you've got a problem, go out and decide whether you need to actually spray your fields or not, rather than just spraying. We can also use these infochemicals to modulate the behaviors of these beneficial insects, this parasitic wasp, which is laying its egg inside a pest. And by changing the chemical environment, we can pull them into the field to increase the effect and thus reduce losses and reduce pesticide use. The other one, of course, is GM crops. Yeah, this one is good, actually, because it doesn't fall off and it fits on a square piece of bread. But there's a great deal of contention about the use of GM crops, although they are being used worldwide. And we currently have a project going on GM crops, not in Canada, but in Brazil, where they grow BT corn. And it turns out that the fall armyworm has developed resistance. And we are looking to see whether the resistance comes at a cost or whether they are actually better or worse than the control ones. And we had a really big surprise. Normally there's a cost. It turns out that we're producing super males and super females when they are resistant to certain varieties, which is going to totally change the way that we do resistance management. And of course, there will always be a cost. And now we're trying to work out exactly how we explain this to all of the sides that love and hate GM crops. Thank you.
Regarding your last image of the super insects, in which way super?
Super in the fact that females spend more time emitting the sex pheromone, the resistant ones. They produce more of the message. And the idea was that normally you would have a lot of non-resistant ones, and a resistant one would most likely be diluted, the resistance, because they would mate with others. Well, they're now having assortative mating, which means the selection will go and we'll get resistance very quickly. However, I should say that in a couple of cases, on other strains, they are actually less fit. But we're getting a bunch that aren't.
This isn't my area, but it's something I've been reading a bit about. How much is the recent research of the epigenome filtering into this kind of research?
With people like me, very little, but there are a group of people that are really considering this aspect, and we have to take a much broader view of what we're doing right now than what we have been doing in the past, no question.
Thank you very much. That was quite impressive. Thank you. I wonder if you can use your pheromones to contain epidemics, for example, like the Zika virus, or use it for malaria control.
There are possibilities in certain insects, but not, the mosquitoes aren't very good. They use pheromones more for choosing where they will lay their eggs and so on. You could possibly use it for that type of management. One of the problems is it works in the lab, getting it to work in the field all of the time, because it's not only monitoring. One they have is called mating disruption, where you put so much out in the field that basically when the female goes, I'm over here, the male can't find her because there is so much in the air. But it only works in certain species. Others it doesn't work at all. They still find ways of doing it. Nature's very clever. Yes.
I was curious about how you actually trap these things. I was at a Microsoft event recently in which they, quote, demonstrated a trap downstairs with live mosquitoes in it. Don't worry, the mosquitoes can't get out. So I'm kind of curious what sort of technology you're using to collect these sorts of insects in ways theoretically.
Well, the light trap is very simple. I mean, if you go out in a cottage and you turn on a light late at night, you see the insects come around. Well, when they fly, they do a flight getting ever decreasing circles. So you have baffles, they bump into it, they fall down into a funnel, which makes it very difficult for them to get out. The pheromone traps are just glue. But, I mean, most of them are set up in a way where they get in, but they cannot get out. A lot of them will put in a compound that will kill them. But if you're like me and you want them live to establish colonies, then you have to have live traps. and you have to make it sure that they cannot get out. But don't ever believe anybody when they say a mosquito won't get out. Sorry, they get out. They get out of my traps too, so I mean, I'm not blaming mosquito people.